Welcome, curious minds, to a journey through the intricate realms of life's mysteries. In this video, we embark on an exploration that unravels the delicate dance between environment and chance in determining the sex of organisms. Are our destinies written in the language of genetics, or does randomness play a pivotal role in the grand tapestry of life? Join me as we delve into the captivating world of sexual determination, where nature's secrets unfold in a mesmerizing interplay of design and serendipity. In all organisms, beyond the sets of genes required to develop both types of gonads and sexual organs, additional selector or master genes determine which of the two sets of genes actually is being used in developing an individual's sex. Typically, one decisive master gene is present but allotted to only one half of the offspring. This is referred to as genotypic sex determination. Alternatively, master genes are activated differentially by environmental influences. Such cases are subsumed under the heading environmental sex determination. In environmental sex determination, the decision is made only after fertilization, and external cues such as temperature or presence of a sexual partner make the choice, presumably by influencing the activity of master genes. For instance, in the annelid Ophryotrocha purilis, two individuals that encounter each other by chance influence each other by means of chemical signals or pheromones, and eventually one individual plays the role of the male and one plays the role of the female. Environmental sex determination has been discovered in several invertebrate species, but also is known to occur in fish and reptiles. Environmental sex determination is indicated but not proven by sex ratios deviating significantly from 1:1. In genotypic sex determination, the decision is left to chance in fertilization. Traditional belief offered by ancient writers such as Aristotle ascribes sex determination, even in humans, to non-genetic determinants such as nutrition or the heat of passion during intercourse. How such factors ensure a sex ratio of 1:1 usually is not the subject of much attention in superstitious explanations. However, how can chance yield a ratio consistently close to 1:1? The principle may be explained using humans as the example. All humans carry 2 into 23, that is 42 chromosomes in the diploid cells. Of these, 2 into 22 chromosomes are indistinguishable in males and females and are called autosomes. In females, the 44 autosomes are complemented by 2 more homologous chromosomes, the X chromosomes. In males, 2 morphologically distinct chromosomes are found. One is an X, like those found in females, while the other is a modified X termed the Y chromosome. X and Y chromosomes are often called sex chromosomes, but this designation is highly misleading. A better but still misleading expression is sex determining chromosomes. With respect to the 2 non-autosomes, in humans and other mammals, females are homogametic with XX, while males are heterogametic with XY. This suffix gametic suggests consequences in gametogenesis. In birds, incidentally, females usually are heterogametic and the males are homogametic. When meiosis takes place during spermatogenesis and a haploid set of 23 chromosomes is parceled out to each future sperm cell, this set includes, besides 22 autosomes, either the X or the Y. As a consequence, 50% of the sperm cells obtain the X chromosome by chance, while the other 50% obtain the Y chromosome. In oogenesis, all future egg cells obtain besides 22 autosomes and X chromosome. In conception, it is a matter of chance whether the sperm cell carrying X or a sperm cell carrying Y fertilizes an egg cell. On the average, Y carrying sperm appear to be slightly faster in the race to the egg than X carrying sperm. Hence, the ratio of 105 males to 100 females. Unexpectedly, intensive research into the nature of genotypic sex determination has shown that the role of chance in distributing chromosomes or genes is the only common denominator among the various types of genotypic sex determination identified in the animal kingdom. Despite nature's tendency to retain established solutions to biological problems, and despite the universality of sexual dichotomy, there is no universal molecular mechanism of genotypic sex determination. In the simplest case, sex is determined by a gene that occurs as two alleles. One sex is heterozygous, the other homozygous. This mechanism occurs in the common housefly Musca domestica. However, in most animal species, the mechanism is more complex and includes cascades of mutually dependent events. Frequently, the sex-determining genes occur in two morphologically different chromosomes, usually called the X and Y. In such cases, one of the two sexes is XX and the other is XY. Despite the consistent XY formalism, the roles of these chromosomes vary in different organisms. In Drosophila, for example, the ratios between the number of X chromosomes and the two sets of autosomes AA in each individual diploid cell is decisive in determining the sex of each individual cell. XX to AA, a ratio of 1:1 is a female, while X to AA, a ratio of 1:2 is a male. The Y chromosome in Drosophila is of significance in spermatogenesis in the adult male but not in early sex development. Sexual determination is cell autonomous and is not mediated by hormones. If one X is lost due to a faulty mitosis or non-disjunction in an XX embryo, the resulting X0 cell and all its descendants become male in female surroundings, while the descendants of the corresponding XXX cells are super female. In insects, unlike vertebrates, there are no sex hormones that could moderate differences in X chromosome numbers and ensure a uniform phenotype. An individual with an abnormal ratio of XX to AAA develops an intersexual phenotype with genetically uniform cells but an intersexual organismal appearance. In Drosophila and other insects, individuals composed of a mosaic of female and male cells are called gynanders, in Greek meaning wife man, or they are also called gynandromorphs. In each somatic cell, X-linked genes code for factors aptly called numerators, while genes located in the autosomes code for denominators. In the presence of two X chromosomes, more numerated proteins are produced by genes that apparently escape dosage compensation or before dosage compensation is accomplished. When numerated proteins predominate, the master gene SXL is switched on. This sex-lethal protein directs a cascade of biochemical events. These include the correct splicing of its own pre-mRNA, by which sex-lethal makes it possible to maintain its own synthesis. The cascade of events set going by sex-lethal terminates in the activation of female-specific genes. In contrast, male development is the default program resulting from insufficient levels of sex-lethal protein to trigger female development. In mammals, the situation is the reverse. The Y chromosome contains a male determining gene and the female development is the default option when the gene is absent or defective. As we conclude our expedition into the enigmatic world of sex determination, one thing becomes clear. The intricate tapestry of life is woven with threads of both design and chance. Nature, in its infinite wisdom, presents us with a fascinating puzzle where environment and randomness collaborate in the creation of diverse and remarkable organisms. Our journey may end here, but the questions linger. Is it fate or fortune that shapes our biological destinies? The dance between nature and chance continues, leaving us with an ever deepening appreciation for the awe inspiring complexity of life's grand design.